Hi everyone, this screencast is going to cover mitosis in the cell cycle. Please make sure you've got a pen and some paper before we start. The LO for today's session is to understand how cells divide. Once you have written down the LO for today's screencast, please have a go at this retrieval practice from the previous lesson on chromosomes. All you need to do is match the keyword to the right definition. Okay, when you're ready to mark your answers, please make sure you've got a different coloured pen, preferably green. So we've got a gene is a short section of DNA which controls characteristics. We've got a chromosome is a long strand of coiled up DNA. The nucleus is where the genetic information is found. And the DNA is a unique chemical molecule. The success criteria for today's screencast is to identify the roles of mitosis, explain the process of the cell cycle, and complete calculations related to the cell cycle. So a quick recap before we get on to the cell cycle. The human cell nucleus contains 46 chromosomes in 23 pairs. They are ultimately long strands of coiled up DNA. DNA has short sections called genes, like you can see here. And these genes control a characteristic of your body. It's important to remember that characteristics are a result of many genes working together. So I've got a couple of quick recap questions just to go through before we get on with the rest of the screencast. So pause the video and have a go at these. Here's the first question. And here's the second question. Make sure you pause the screencast. I'm going to go through these in a second. So for the first question, it tells us that a sperm cell from a dog contains 39 chromosomes. And the question asks us how many chromosomes there would be in each dog muscle cell. We know that sperm cells are haploid, so they have half the number of chromosomes that a body cell would have, and a muscle cell is a body cell. So we know that the muscle cell must have 78 chromosomes. For the second question, how many pairs of chromosomes are there in a body cell of a human baby? We know that in a body cell of a human, there are 23 pairs of chromosomes. We've got to be careful with these questions. If it's asking us about the number of chromosomes, our answer will be 46. But when it asks us about the number of pairs of chromosomes, then our answer is 23. Then we have to put the following in order of size. We're starting with the smallest for number 1 and the biggest will be number 4. So first we have the gene, followed by the chromosome. Remember, a gene is a section of a chromosome. We've then got the nucleus, which contains the chromosomes, and the cell, which contains the nucleus. So cell division then, cells are continually lost and made. To make new cells, the body needs to carry out cell division, and it does so in a process known as the cell cycle. Cells divide where the body needs to either grow, replace worn out cells, or as you can see here, replace damaged tissue. So the role of mitosis is really important for the growth and development of multicellular organisms. We are multicellular organisms, so I'm using humans as an example here. When a sperm cell fertilizes an egg cell in human reproduction, the resulting zygote cell forms and divides by mitosis. So a zygote cell is a fertilized egg cell. So we could have this as an example here. It goes all the way through the cell cycle to form 2 new genetically identical daughter cells. These cells will then go all the way through the process again and form more and more cells as the process goes on. And eventually, we've got enough cells to have formed an embryo. As the embryo continues to grow in size, so we've got the cells dividing by mitosis, and they start to differentiate, we have a fetus forming. So we have those specialised cell types starting to form to create a fetus. So we've looked at the roles of mitosis. Now we're going to explain the process of the cell cycle. So before we go on to the cell cycle, I just want to have a look at the units that we use to measure the genetic material. So we use a unit called picograms, and a picogram is 10 to the power minus 3 nanograms. So if we look here in this table, we've got 1 picogram is 0.001 nanograms. I've put this table in, one, to show you the scale. So picograms are 1 times 10 to the minus 12 grams. So they are very, 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 very small units of measurement. This table will be helpful for you to copy down. It will be helpful in this screencast, and it will also be helpful if you can revise this for your exam. I have put on the sums that we would do to convert between units. So if we were going to go from nanograms to micrograms, for example, we would divide by 1,000. If we were going from micrograms to nanograms, we would times by 1,000. So mitosis is a type of cell division, and it's where the cells produce identical copies of themselves. It's used for growth, repair, and asexual reproduction. It differs from meiosis. Meiosis is the process that gametes divide by. So it's the type of cell division that produces gametes or sex cells, so sperm cells and egg cells. The cell cycle as a whole consists of 4 distinct stages, and mitosis is one of those stages. We have a growth phase, which is made up of 3 phases, and then we have mitosis. So the first phase of growth phase is gap phase 1. So the cell grows bigger, it replicates its organelles, so the number of ribosomes increase, and this helps the cell to build proteins. And the number of mitochondria also increases, and this provides the cell with energy for cell division. Then we have the synthesis stage. The cell replicates all of its DNA to form 2 copies of each chromosome. And then we have gap phase 2, which is where the cell keeps growing until all of its organelles have duplicated. So if we summarise the growth phase, all 3 phases, it involves cell growth, DNA replication, and the numbers of subcellular structures increasing. So if you now move on to mitosis, it's the process where 1 set of chromosomes is pulled to each end of the cell and 2 new nuclei are formed. At the end of mitosis, there is a step called cytokinesis, and this is where the cytoplasm and the cell membrane divide to form 2 identical cells. In your exam, you'll be shown a simplified cell cycle with usually 3 stages. So the first stage will be growth phase. This will involve cell growth, DNA replication, and the numbers of subcellular structures increasing. Then we will have stage 2 as mitosis, where 1 set of chromosomes moves to each side of the cell and the nucleus divides. Then we've got stage 3, which is cytokinesis. This is where the division of the cytoplasm and cell membrane happens to form those 2 new cells. Another exam tip is that you might be expected to look at micrographs or images from a microscope and identify different stages of mitosis. So you'll have to be able to recognise and describe what is happening in those images. Cells that aren't actively dividing should be easy to identify because their chromosomes will be unravelled. So they'll look non-distinct or they'll appear like strands of thread. So I'm going to show you what the different stages of cell division would look like under a microscope. If you do want to copy these down, you will have to pause it. So this is what the cell looks like when it begins to divide. So we can see the chromosomes, they're very distinct. Then DNA replication occurs, so they've all replicated and the nuclear membrane has broken down. Then the chromosomes line up along the equator or the middle of the cell. Then we have spindle fibres forming, and these attach to the chromosomes. These spindle fibres contract or shorten, and this pulls the chromosomes to opposite ends of the cell. So remember, we've got 1 of each chromosome because they replicated. Then the nuclear membrane reforms, so we've got the 2 nuclei forming here. And then cytokinesis occurs and we have the 2 identical daughter cells being produced. So I've got 2 exam questions for you now. I want you to pause the video and have a go at them. So here is the first question. And here is question 2. When you're ready, we'll go through the answers, so make sure you pause it if you need more time. For the first question, this is a kind of simplified cell cycle you will see in an exam. It tells us here the stage 2 here is mitosis. So we need to draw one line from each stage in the cell cycle to show what's happening during that stage. So we know that stage 1 comes before mitosis, so it must be growth phase. And this is where copies of the DNA are made. Stage 2, it tells us, is mitosis, so we know the nucleus is going to divide. Then stage 3 must be cytokinesis, and this is where the cell divides into 2. For the next question, it's asking us about the mass of DNA in a human body cell. So at the start of the cell cycle, it's 6 picograms. It's asking us what it will be in the new cells that are produced. This is mitosis, so we know that the cells produced are going to be genetically identical to the original cell. So it will be the same mass, so the answer is 6 picograms. So I'm now going to go through the final success criteria, which is to complete calculations related to the cell cycle. So you might be asked to use observations of the number of cells in each stage to calculate how long each stage actually lasts. So this is an example of one of those questions. So the student is told that the whole cell cycle takes 15 hours and they're asked to calculate the length of time that stage 1 lasts. So if we look at the number of cells that are in stage 1, we can work out the proportion if we divide that by the total number of cells. So we do the observed number of cells in that stage divided by the total number of cells, and we then times that by the total length of the cell cycle, in this case 15 hours. So our sum is going to be 14 divided by 54 times by 15, that gives us 3.88 hours, or 233 minutes if we multiply that by 60. You might also be asked to calculate percentages of the time taken up by one stage of the cell cycle. So for example, for this question, what percentage of the cycle is spent in cell growth? We would look at how we can work this out. So we're going to look at these increments. Cell growth takes up 10 out of a total of 20 increments here. So we're going to do 10 divided by 20, that gives us 0.5. To work out the percentage, we're going to times that by 100. That gives us 50%. If we then want to use the same method to find out how long that specific stage takes, we can work out the proportion in exactly the same way and multiply it by the total time of the cell cycle. That would give us 10.5 hours. So for this question, the total cell cycle takes 21 hours. So I'm now going to put 3 different exam questions on the screen. I need you to pause the screencast on each question because I'm only going to leave them up for a few seconds, and then we'll go through the answers together. So this question is asking us to calculate how many hours are spent in mitosis. We've got a full circle here, and it tells us that the mitosis stage is 28 degrees out of this full circle. So that's going to be 28 degrees out of a total 360. So we do 28 divided by 360, which gives us 0.078. We then times that proportion by 15 because that is the total time taken for the cell cycle. That gives us 1.17 hours. So this question is asking us to calculate how many hours cell division takes. So it tells us that cell division takes 5% of the total time, and the total time is 21 hours. So we're going to convert our percentage back into a decimal. So we're going to do 5 divided by 100, which gives us 0.05. We're then going to times that by 21, which is the total time. That gives us 1.05 hours. For this question down here, it's asking us what percentage of time is spent copying the DNA in the cell cycle. So there's 7 of these increments out of a total 20. So 7 divided by 20 gives us 0.35. If we times that by 100 for a percentage, then we get 35%. The final question asks us about the 3 stages of the cell cycle, but it is a 5-mark question. So we need all 5 points for 5 marks. We need stage 1, cell growth and DNA replication. Stage 2, mitosis, the chromosomes move to each end of the cell. The nucleus also divides, and then stage 3 is cytokinesis. And there we are done.